Hello, seventh graders learning from home. Um, so what we're going to try and do in this video is walk you through what you would have seen by doing the microscope lab in school. I know this is not going to be perfect, but we'll try and uh, make it so we learn something from this and you get to see some of the things that you would have seen had you been in school to do this lab. So we are doing the using a microscope lab. This lab handout can be found in Schoology. Um, looks like this. You should download that and put it into Notability if you have not already. Um, and I'm going to be stopping periodically and telling you to pause the video and do a couple of sketches and answer a couple of questions as we go. So the purpose of this video was for you to get a chance to use a microscope for the first time. I know that this is not ideal. And I'm sure that later in your school career, 10th grade, maybe even next year, you'll get a chance to use a microscope to look at some rocks next year, possibly. But if not, in 10th grade biology, you'll definitely get a chance to use these. If you have used a microscope before, this may be a little bit of review. If you have not used a microscope before, this is just going to be a chance for you to get a little bit of an idea of how they work. So when we started off in this activity, we went over the steps for making what's called a wet mount slide. And that's going to be a slide that uses water or some type of dye or stain to suspend what you're looking at under the microscope. And so we always want to start with a clean slide. We're going to put the item we're looking at on the slide. You're going to put a drop of water, or if you were using a stain, you would use a stain onto your slide. The slide is just a rectangle of glass generally. Then we're going to put what's called a cover slip onto that slide, and that's usually a smaller piece of glass or it's thinner. We use plastic just because very thin glass is very breakable. And in 7th grade, sometimes things break. We start always with the lowest power objective lens on our microscope, and then we work our way towards high power. And so that's what we're going to be working on here. So we started by looking at a word from a newspaper that had the letter E in it. And so we followed those steps. We put a drop of water on top of it. We put the cover slip on it, put it under the microscope on the lowest power, which is called the scanning power lens. And we got something that looked like this. You're going to be able to see a couple of letters. I think this is a little bit smaller because it was taken with a with a digital camera attached to a microscope. So it's not quite exactly what you would see under the microscope. Under a true microscope, it'd be round. You'd have a larger view. You'd be able to see a little bit more of the word. And so first of all, what I would like you to do is to simply pause this video and make a sketch of this E. Okay. When we put this E under the microscope, it was right side up, just as if we were reading that word on the slide. And this is what the microscope gave us. So pause the video and please make a sketch in this circle right here on your worksheet. Okay, now that you've got that sketch made, we've got a couple of questions to answer. The first one, how is the E in the circle different than the E in the rectangle? So this would be the E in the circle. So what you're seeing under the microscope. The rectangle they're referring to, or I'm referring to in this question, is right here on your worksheet. And so how is it different? Well, hopefully it's obvious. It's upside down and it's backwards. And that's something that you're going to notice in this lab. So all you need to write on this line is the E looks upside down, it's backwards. You may have also noticed, look at all these paper fibers. Isn't that crazy? You can see the lines going across there. And this is the little tiny pieces of wood pulp that are put into paper. Next, we need to look through the scope and pull the slide slowly toward yourself and describe what you see. So I'm going to jump over to the gizmo we looked at when we were learning about microscopes, and hopefully you'll be able to get a chance to see this a little bit better. So this item that we were looking at here is called Elodea. Elodea is a freshwater plant. It's one that we have used in classes. People keep them in their aquariums. You may have it in a river, lake, pond near your home if you live near one of those. And so the first one says, pull the slide toward yourself and describe what you see. So I'm able to move this slide up and down using the gizmo. And if you want to go back into that gizmo and play with this a little bit more, there's a bunch of other different stuff you can look at under the microscope that you may not have looked at when we did the gizmo. If you just want to get a little more of an idea of what a microscope can show you. So we're looking at this freshwater plant, though, called Elodea. It says to pull it towards yourself. So for me to do that, I'm going to pull it down on the stage. So watch what happens as I pull it down. What way does it look like it's going? Here, I'll do it again so you can see. If I'm pulling it down, it looks like it's moving up. And that's what you're going to need to write down. When you pulled it toward yourself, you would have seen that it moves up, like it's going away from you. The next one says, move the slide to the left. Which way does the E appear to move in the microscope? So if I move this slide to the left, what does it do? Does it look like it's moving left? No. It's going the opposite direction. It's moving right. So pause the video, write that down. It looks like it's moving right. Next one says, move the slide to the right. Which way does it appear to move in the microscope? So if I move this to the right, it looks like it's moving left. And that's all you would need to write down. It's just your observation of that. Okay. These questions were really to get you to see that when you're moving things in the microscope, they move the opposite direction. Next, we're going to move on to the next question. And we're going to go back to that newspaper E, and we're going to look at it under high power. And so I've got another photo. This document, by the way, that the photos are in is in Schoology for you to look at on your own if you want to. And so this second photo would be under low power. The first one is called the scanning power lens. Then we've got low power. And then this photograph is one that would have been taken under the highest power. And so all you need to do for this part is to just make a sketch in this circle of what you see. Now you're probably noticing you can't even tell that this is the letter E from a microscope. You can't tell what letter this is. You are so zoomed in on this. All you can see is the paper fibers and some ink blotches. Like you can tell this is probably a curved letter, but in your drawing, you just want to focus on getting some paper fibers and some of the ink on there as best you can. Okay. Make sure you're using the finer tipped drawing tools in Notability. Please don't make this look like you do it with a great big fat marker or a great big fat paintbrush. So pause the video if you need to do that drawing quick. Okay, next we are going to learn about how to figure out the total magnification of a microscope. So to do this, you have to do some multiplying. As it says, to find the total magnification, multiply the magnification of the eyepiece, also known as ocular lens, which says 10x on it, and the magnification of the objective lens. So looking back at this gizmo, just to remind you that the eyepiece or the ocular lens is up here, and these three down here are the objective lenses. And as you change the magnification, they rotate on the revolving nose piece, is what this part of the microscope is called. So looking at these, you need to multiply to figure out the total magnification. So for this first one, the total magnification of the scanning power, it's red on the microscopes we have at school, objective lens, which is 4x. So it magnifies things 4 times, and the eyepiece does 10 times. So to get the total, you just multiply that. Do that math in your head. 4 times 10 is 40. Hopefully you got that. Write 40 down on this first line. Second one, the low power objective lens, that magnifies things 10 times. So for this one, you're taking 10 times 10. Drum roll. You got it. It's 100. Next one, the high power or the blue objective lens magnifies things 40 times. So you figure out the total by multiplying them 40 times, 10 times, 40 times 10. Add that 0, gives you 400 times is your total magnification. Now we're on to part 2. If you were in school, I would have you rinse off your slide, throw your little chunk of newspaper away, and you and your partner in a normal year, I would ask both of you to pluck a piece of hair and make an X with them so you could compare them together. It's really crazy to see how different some people's hair can look. Sorry, pause the video there for a sneeze. So I've got a picture of a piece of hair under high magnification here. But as I was saying, it's crazy to see different people's hair under a microscope. If you've got dark, thick hair, it's going to look something like this one in the picture. If you have darker, thin hair, like me, because I'm old and I'm going bald, it's going to be like half this thickness. If you have blonde hair or reddish hair, it's almost see-through. It's like transparent. The crazy thing is that if someone has dyed hair, or say if this person whose hair we're looking at in this picture had bleached their hair, that bleach or dye only shows up on the outside. So if this person had bleached their hair, the inside would still be kind of darker colored. If you had dyed hair, so you have light hair and you dyed it darker, the dark color would be only on the outside in your hair if you had dyed hair. So what I need you to do for this part is you're going to make a sketch of this, what you see. Pause the video and do that. Now that you've made that sketch, there's one question that goes along with this. Where is the color found in each hair? So is it spread all throughout the hair? Is it only in the center? Is it only in the edges? Where is that color located? And just write that down here. It's your observation. For this last part, we've got a couple of questions for you to say which direction you have to move things, and then we've got an I learned question. So for these 4 circles down here, you are imagining that you're looking through a microscope and you need to get this E in the center of your view. So remember that it always moves the opposite direction. So I'll do this first one with you, and then you can do the other 3. So for this first one, to get this E in the center, I would have to move it down and to the left. So remember though, it moves opposite. So what I would write on this line is I would need to move it up and right to get that E centered. So you do these next 3. Remember that it moves the opposite direction of the way you move it in the slide, or under the microscope, your slides. To get that E centered, right direction, then you would have to move this. And then lastly, in this lab I would like you to just write down something you learned in this lab. I know and I completely understand that this is not the same as if you were in school, but hopefully you got a little bit out of it, and just a little introduction of how microscopes work. Have a good day. Let me know if you get stuck on anything or you have any questions, and I'll be glad to help you out. We'll have some Google Meets open this week, as always, during 3rd hour. And if you can't make it to one of those, send me a Schoology message. I'll let you know. I'll get back to you as soon as I can. Thanks. Have a good day.